So welcome back guys. This video is going to be about the genetic code and amino acids. So basically the genetic code is a triplet in nature, like we all know. So as there are 4 nucleotides A, G, U, C, this number will be same as there are 20 amino acids. So we want something that covers them all, so that's why it's like 4 raised to the power 3. So it's a triplet that contains total of 64 codons, and that are coding are 61, and the ones that are non-coding, the stop codons, those are 3. And if genetic code was quadrant, it will be like 4 raised to the power 4, so 256 codons if the genetic code was quadrant. So the genetic code was initially assumed to be unambiguous in nature, that is, one codon is specific for a particular amino acid only. But actually the genetic code is degenerate in nature, that is, more than one codon can also code for the same amino acid. So they are called as synonymous codons. Different codons which code for the same amino acid, example UUU, are also coding for phenylalanine, and so does the UUC. So that's the meaning of synonymous codons. So you can learn this like leucine, serine and arginine are like coding 6 codons. Okay, so number of synonymous codons for leucine, serine, arginine are 6. For isoleucine is 3, methionine and tryptophan is 1, and AUG is for methionine. For glycine, proline, alanine, valine and threonine, it's 4 each, and for the rest of them it's 2. So now there can be a question like how many different mRNA are possible for the following polypeptide. So you can say there is isoleucine, and the number of synonymous codons that it codes is 3. So 3 into methionine is 1, into 2 into glycine and valine are 4, into 4 into 4 will give you the number of mRNAs that are possible for this following polypeptide. So now the 3 stop codons are UAA, UAG and UGA, but they can sometimes be coding in nature, like UGA may code for selenocysteine and UAG may code for pyrolysin. Now selenocysteine and pyrolysin are amino acids which are derived from enzymatic modification of the standard amino acids. Selenocysteine is present in case of eukaryotic enzymes like formaldehydrogenase and glutathione peroxidase, but the function for pyrolysin is not yet described. So the first codon of every mRNA is called as the initiation codon, and it can have 2 possibilities in case of prokaryotes and eukaryotes. So in case of prokaryotes, the initial codons are AUG, GUG and UUG. So when it is formulated with methionine, it's the initiator amino acid for prokaryotes, and for eukaryotes you may know that it's AUG and it's normal methionine, the unformulated form. So that is the initiator amino acid for eukaryotes. Okay, and the selenocysteine amino acid is derived from enzymatic modification of serine. Okay, the name might suggest you it comes from cysteine, and the structure may also like like I'll show you the structure later. It's just cysteine and there is selenium at the end. So but actually selenocysteine is derived from the enzymatic modification of serine containing tRNA, and CSLB is an elongation factor that is involved. So now you can learn the symbols for like the amino acids, and basically only BJOUXZ, BJOX is how I pronounce it, just to remind me, like for me to remember it for later. BJOX, BJOUXZ are the ones that do not like are not the symbols for any amino acid, and others you can just write A for alanine, and ALA is the short form, C for cysteine, D for aspartic acid, E for glutamic acid, F for phenylalanine, G for glycine, H for histidine, I for isoleucine, K for lysine, L for leucine, and M for methionine, N for asparagine, and its like symbol is ASN, and P for proline, and Q for glutamine and its symbol is GLN, and for glutamic acid it was GLUE. Okay, and R is for arginine, that is ARG. So know the difference between ARG and ASN. Okay, asparagine and asparagine, it's like short form is N. Okay, like that's the symbol for it. Arginine, R, arginine, so it's like R. And S is serine, T is threonine, V is valine, W is tryptophan, Y is tyrosine. For T it's threonine, just remember, and for tryptophan you remember it's W, and for tyrosine it's Y, because tyrosine, okay, that's how you can remember. And now coming to the genetic code, you know that the first position, second position, third position. First position is always you write like UCAG, UCAG, UCAG. So U is for phenylalanine, U. Okay, and before already I taught you like phenylalanine already has, like, it came in the rest, so it has how many number of synonymous codons? 2. So, and also you can learn this just in case it comes in some form of question. It's just very simple. First 2 are phenylalanine, then leucine, all 6, and then isoleucine, then 1 methionine. From 1 methionine, you remember tryptophan and methionine just had 1, and then valine, and all 4 valine, or alanine, and serine, proline, threonine, adenine. Septa is how I like learned it, SPTA, septa, serine, proline, threonine, alanine. And then 2 tyrosine, and UAA and UAG and UGA are stop codons, so stop. And then tryptophan, like 1 only number of synonymous codons, so only 1. And then histidine, glutamine, and asparagine, lysine, aspartic acid, glutamic acid, arginine, and then both serine, and then again 2 arginines, and then glycine. Okay, that's how you can like remember it. So before seeing the structures, I might show you this, the classification of amino acid based on chemical nature of R group. So basically aliphatics are glycine, alanine, valine, leucine, and aromatic are phenylalanine, tryptophan, and tyrosine. Hydroxyl are serine and threonine. You will see that in their structure they will have OH. And carboxylic is for aspartic acid and glutamic acid. Sulfur containing are only cysteine and methionine. Amino means amino group of any molecules which combines with carboxy group of same molecule is proline. Like see in this structure, proline has amino group attached to the same carboxy group of the same molecule, so that is amino, that's proline. And amino is lysine and arginine, and amide is glutamine and asparagine, and amides are R, O, N, and any 2 groups. So basically stop codons are UAA, UAG, UGA, and amino acids with non-polar side chains, that is hydrophobic. And also the most hydrophobic is isoleucine. Okay, so the non-polar ones are glycine. Structure is very like after the zwitterion you can see like it's just H, COO, NH3, CHH, CH3 just add CH3, it's alanine, and then CH, CH3, CH3, valine, and then just add CH2 in between, becomes leucine. And C, CH3, CH2, CH3 is isoleucine, which is the most hydrophobic one. Proline, which is amino, it has CH2, CH2, 3 CH2s, and the last one is attached to NH3, that's why it's an amino. Methionine has like 2 CH2, CH2, and as I said, cysteine and methionine are sulfur containing, so there is the sulfur and CH3. Okay, CH2, CH2, S, CH3. Phenylalanine, tryptophan are both like aromatic, like we learned, and tyrosine also we'll see. So here it's just CH2 and benzene ring. Tryptophan is C double bond CH, NH, okay, 5 ring, and then a benzene ring. So also you can see non-polars like the ones I just discussed, glycine, alanine, valine, leucine, isoleucine, proline, methionine, phenylalanine, and tryptophan tends to like go inside. Okay, and they are mostly hydrocarbons. Polars like acidic, which are like negatively charged, are having COOH group, and they are aspartic acid and glutamic acid. D is for aspartic acid and E is for glutamic acid, and the basic ones are amino, arginine for R, K for lysine, H for histidine, And the polar which are uncharged, neither they are amide or alcohol like the hydroxyl one, serine and threonine, and cysteine having the S. And polars tend to face outside. That's why the polar ones are hydrophilic. Okay, they love water. That's why they can face outside. They don't tend to go inside like hydrophobic non-polar. And the most hydrophobic one is isoleucine, and the most hydrophilic one is arginine. Okay, so now amino acids with uncharged polar side chains at pH 7 tends to face outside, and the serine and threonine are hydroxyl containing. See the structure OH, OH, H. If CH3 is there, it's threonine. It's very easy to learn the structures. You just have to understand the basics. Like this is same always, and just CH2 and then SH is cysteine. And asparagine has like C double bondo amide, C double bondo NH2. Okay, and again glutamine is also a amide, C double bondo NH2. Only difference between asparagine and glutamine is glutamine has 2 CH2. Tyrosine is again aromatic, so hydroxyl aromatic, CH2, same as phenylalanine, just add the OH. Okay, so now the ones with the charged polar sides are 3, which are basic positively charged, are lysine, arginine, histidine. You can see the like structure 1 2 3 4 CH2, NH3 is for lysine, and then 3 CH2, NHC, NH2 plus NH2 arginine, and for histidine there is this ring. Okay, 5 1 2 3 4. And for aspartate and glutamate, they are negatively charged, so they are acidic, and the structure is also like simple, like they will have, because they are acidic, COO. Aspartate has CH2 and glutamate has CH2, COO. Okay, so basically these are the 21st and 22nd amino acids which are derived after the enzymatic modification of the normal ones. So just for the structure, and like you can learn the structure, I have like made the cysteine here. So basically it's simple, same like cysteine and then just selenium and H. But do remember that it's the enzymatic modification of serine. CSLB is like one that is involved, the elongation factor that is involved in this conversion. Okay, pyrolysin is kind of same lysine. Okay, like lysine in till here, like 4 CH2, 1 2 3 4 CH2, and then NH, C double bond O, and 5 ring and CH. So there are like 7 essential amino acids: lysine, methionine, leucine, isoleucine, valine, tryptophan, and phenylalanine. And in humans, threonine is also an essential amino acid. Arginine and histidine are semi-indispensable, like they are slow to be formed in humans, and the rest are non-essential. So basically there are 2 types of peptidases, like the enzymes for protein digestion: endopeptidases and exopeptidases. Endopeptidases are pepsine, chymotrypsin, trypsin elastases. They cleave internal peptide bonds. And exopeptidases are the ones that cleave amino acid either from carboxy, COH terminal, so-called carboxypeptidases A and B, or from amino terminal, so-called aminopeptidases. So like pepsin cleaves from acidic or aromatic amino acid. Okay, the pepsin cleaves peptide bonds in amino terminal side of the cyclic amino acid residues. Cyclic amino acid residues are the same as aromatic, that's what they mean: tyrosine, phenylalanine, tryptophan. Pepine cleaves at phenylalanine, arginine, and lysine. You may find a lot of questions from these, like where do trypsin cleave, or they'll just give you like A, R, S, D, S, A, and they'll ask like where will trypsin cleave. So you have to remember trypsin cleaves at C terminal of arginine or lysine. Lysine is K, arginine is R. The symbol you have to remember, all that is taught in this video to like answer the questions regarding this. So chymotrypsin, they cleaves at tryptophan, phenylalanine, tyrosine, leucine. Elastase cleaves at alanine, glycine, serine. Do remember guys, like do remember these, because I know I confused elastase with chymotrypsin and I like got an answer wrong. So remember, elastase is alanine, glycine, serine. Chymotrypsin is tryptophan, phenylalanine, tyrosine and leucine. Okay, all of the aromatic plus leucine are cleaved by chymotrypsin. Pepsin, acidic and aromatic, and cyclic ones. Aromatic, obviously same. Okay, and then papain, phenylalanine, arginine, lysine, trypsin, arginine and lysine, C terminal of arginine and lysine. Now coming to the exopeptidases, carboxypeptidases A and B. A1 cleaves at hydrophobic amino acids and B cleaves at basic amino acids. Basic ones were arginine, lysine and histidine, right? So mainly arginine and lysine is where carboxypeptidases B cleaves, and carboxypeptidases A cleaves hydrophobic amino acids. And just an extra question that I have is, soybean oil has glycine, alanine, serine, not methionine. What does soybean oil already has in it? Glycine, alanine and serine amino acids. And one more important one are the water hydrophobic and water hydrophilic. So these are the symbols for hydrophobic ones. Once you remember the symbol, it will be very easy for you to remember. GAV, GAVLI, MEMP, WYF are the hydrophobic. You have to remember GAVLI, MP, WYF. W was tryptophan, remember. You have to first remember the symbols. And then Q, C, S, T, N, H, R, K, D, E are hydrophilic. The most hydrophobic one is isoleucine. The most hydrophilic one is arginine. All the best. Thank you.